This is the story of how two maverick scientists, a brash American biologist and a boisterous British physicist, cracked the code of life itself. It's a tale of towering ambition, humiliating failure, a flash of stolen insight, and a discovery that would forever change our understanding of what it means to be human. This is the race to uncover the secret of DNA. The year is 1951, and the hallowed halls of Cambridge University are buzzing. The greatest puzzle in biology, the structure of deoxyribonucleic acid, or DNA, is waiting to be solved. Enter our protagonists. James Watson, a restless 23-year-old American with an almost reckless ambition, and Francis Crick, a brilliant, talkative 35-year-old physicist who had switched fields to study life's deepest mysteries. While other research groups, like the one at King's College London, were painstakingly conducting lab experiments, Watson and Crick's approach was unorthodox. They weren't mixing chemicals. They were building. Armed with metal stands, wires, and tin plates, they were attempting to assemble the molecule of life like a child's toy, relying on logic, intuition, and sheer intellectual force. Their ambition was matched only by their impatience, which led to an early and embarrassing misstep. Convinced that DNA was some form of helix, they hastily constructed a model. It was a bold attempt, a triple helix with the phosphate backbone tucked on the inside and the bases pointing outwards. They confidently presented their creation to a delegation from King's College, including the meticulous X-ray crystallographer Rosalind Franklin. The presentation was a disaster. Franklin, a far more rigorous chemist, immediately tore their model apart, pointing out that it contained a fundamental error regarding its water content. It was a crushing, public failure. Humiliated, their superiors at Cambridge ordered them to abandon their work on DNA entirely. But the race wasn't over. The story took a decisive and controversial turn. A short time later, Watson visited King's College. There, Franklin's colleague, Morris Wilkins, working separately and often at odds with her, showed Watson something that would change everything. It was an X-ray diffraction image of a DNA molecule, an image of breathtaking clarity that Franklin had captured months earlier. Known to history as Photograph 51, it was the vital clue. For Watson, it was a moment of pure epiphany. The distinct X pattern screamed helix, and the spacing of the dark smudges on the film allowed him to instantly calculate its dimensions. In that single glance, he saw the truth. It couldn't be a triple helix. It had to be a double helix. Watson raced back to Cambridge, his mind ablaze with the new data. He and Crick, now working in secret, furiously began reassembling their model. With the precise dimensions from Photograph 51, the pieces finally started to fit. They constructed two sugar phosphate backbones, spiraling around each other. Then came the final stroke of genius, the rungs of the ladder. They realized the four chemical bases adenine A, thymine T, guanine G, and cytosine C must face inwards, pairing up in a specific, complementary way, A always with T, and C always with G. It was elegant, it was beautiful, and it explained everything. They hadn't just found the shape of DNA. They had uncovered the very mechanism of heredity, the secret of how life copies itself. Their discovery, published in a short paper in 1953, ignited a revolution. It marked the birth of modern genetics and the entire field of biotechnology. Watson, Crick, and Wilkins would later share the Nobel Prize for their work, a recognition that controversially omitted Rosalind Franklin, who had passed away. From that small model of wire and tin, an entire new era of science was born, fundamentally reshaping our world and cementing the legacy of the two men who solved the ultimate puzzle of life.